Welcome back, dear audience. I hope you all have by now found your clarities on so many basic immunology concepts through these insightful podcasts by SKA. Nevertheless, the journey has just begun. As we venture ahead into the magnificent depths of this intriguing subject, we just come to realize how much of our daily and adult life depends on the build of our innate immune system. The findings have never ceased to surprise even the greatest of experts. It is this factor of understanding something new and better every time we think and look more comprehensively that drives the passion towards this field. So let's keep learning and make aware the upcoming generations of healthier and safer choices. Keep sharing these podcasts with your friends and colleagues, and do not hold back from sharing your views and queries with us. Your host, SKA Talks.
Hello, my friends. Welcome back to the podcast once again, Immunology for Everyone. And I'm sure you all are doing very well. We all know very well that we all have a robust immune system which gets activated on exposure to a pathogen or cancer, and on activation it produces certain chemicals and substances which help our body to eliminate the causative agent. But today, uh, I want to talk to you about a condition when a person's immune system it gets activated to some substances in the environment which are generally harmless to the people in general, and this condition is known as allergy. And we all have experienced the symptoms like sneezing, wheezing, coughing, a runny nose, itchy eyes, which are also some of the main common symptoms of an allergy. However, frequent episodes of this kind of symptoms can make our lives quite miserable, especially during some seasons, which are also called the allergy seasons. This terminology is used because the concentration of some of the allergens like pollens, grasses, and weeds is very high in the atmosphere during these seasons. But there are some allergens which remain all around the year. These are kind of dust mites, pet dander, droppings of cockroaches or mouse. They present in the household all the time, and they may keep causing allergic symptoms in some individuals who are predisposed, or they are susceptible to the allergies. And you know that the children are the hardest hit among all of the individuals because of their immature immune systems, and they can start experiencing such episodes very early in their life, as early as when they are only 2 years old. So let's see what is an allergic reaction. The allergic reaction is defined as the way our body responds to an allergen. It's the simplistic definition. And if you have allergies, the first time you encounter a specific allergen, your body responds and your immune system starts making antibodies against that allergen. And in allergic conditions, we generally find abundance of IgE type of antibodies. And why do IgE type of antibodies are specifically causing allergic reaction? Because these IgE antibodies, they basically can go and bind to a specific type of cells which are called the mast cells. And these mast cells have got the receptors, the Fc receptors on their surface for anchoring of the IgE type antibodies. And once these IgE antibodies they bind to antigen on their antigen binding site, the allergen, and then they anchor onto the mast cell. This basically leads to a passing of signal, and which releases certain granules from these mast cells which carry a lot of histamine. And histamine is the cause of symptoms like allergic rhinitis, running nose, rashes, itching, trouble in the breathing due to the constriction of airways, impaired hearing sometimes, and even abdominal pain. So there are so many symptoms which can be included in the allergic reaction. And the allergies increase a risk of asthma, because both allergies and asthma are conditions mediated by immune activation and inflammation. And for your information, asthma is a chronic condition that causes inflammation and narrowing of bronchial tubes. These are little passageways that allow the air to enter and leave your lungs. And that's why asthma is common in children who are prone to the allergies. So how can we manage the allergies? Managing allergies can be sometimes very tough, because sometimes these allergic reactions can be life threatening. Although there are several allergy remedies that can be used to prevent the symptoms, and but the best remedies to avoid the contact with a substance that trigger the allergy. So every person may have a different kind of allergy to different substance, and it has to be detected uh, what a person is allergic to. And if that person avoids that allergen, that is the best remedy to prevent the allergic reaction. And besides, the doctors may prescribe certain daily intakes of antihistamines or other medications, which can also control the symptoms and prevent or may reduce the recurrent allergic reactions. So the frequency of allergic reactions can be reduced by using. And then the preventive allergy remedies also include keeping your house clean, vacuuming carpets to remove the dust, and pet dander inside your house, using air purifiers, and changing the air filters regularly. So all of these, they basically help in reducing the allergens in your house, and that may reduce the frequency of allergic episodes. Now, why do some people have allergies and asthma, and why not the others? So, a general understanding is that the children who have allergies and asthma, they have some kind of family history, which indicates that the allergic conditions are basically related to genetic makeup of an individual. And this statement is basically true to large extent, but it is not absolutely true, because many people develop allergies even when no known person in their family has ever had such condition. So one should ask a question: what can be other reasons for developing allergies? And in especially in those individuals who do not have a family history. So this question was also asked by a group of scientists from Australia, Switzerland, and Poland, and they conducted a collaborative study. And in that study, they basically used some experimental animals like mice, and they conducted a systematic study, and they got very interesting results and very significant findings, which they published in a very reputed journal, the Immunity, which is published in August 2024, only very recently. So let's try to understand what was this study, because once we understand that, you will get to know what are basically repercussions of having these allergies, and how they develop in children, those who do not have a family history. Uh, in that context, you might recall that While I was discussing innate immunity in one of few earlier episodes of my this podcast, I talked about microbiota when I was talking about innate immunity. And you know that this is a normal microflora of our gut and all mucous membranes in our body. And we also discussed that it plays a very important role as a physical barrier, because it protects the invasion of pathogenic bacteria in our gut, and so is a part of our innate immune system. And there I also mentioned that the excessive use of antibiotics that leads to the elimination of pathogenic bacteria, of course, but also depletes the microbiota, and it causes dysregulation of immune mechanisms. And interestingly, the research study which I am referring today is also relates to the ill effects of use of antibiotics very early in in the life of children. And it revealed that the use of antibiotics early in the life causes some long lasting defects which lead to the development of allergies and asthma in these children. So in order to understand the mechanisms of long term consequences of allergic treatment, antibiotic treatment, these authors they set up a experiment on mice. So they took the very young pups of only 3 weeks old, and they gave them antibiotic in their water for 4 weeks. Uh, and after 4 weeks, what they did was they took the mice and they co-housed them with the similar colony, similar strain of mice which were not untreated, which were not treated. So you have the when they are co-housed, they live together in the common atmosphere. There is a normalization of microbiota. You know, when you have a person whose microbiota is depleted, if you put them in an atmosphere in those in the vicinity of those people who have normal, so there is a normalization. So. Uh, that's what they found. After only 6 days of co-housing, they found that the bacterial density, the diversity, as well as the load was almost restored in the antibiotic treated mice pups. Uh, and they found no significant difference in the abundance of bacterial taxa when they checked after 21 days of housing, co-housing, which indicated that antibiotic treatment had no significant effect on the lung microbiome uh, uh, in that. So there was a normalization after that. However, they kept on co-housing, and after 40 days of co-housing, it was found that the antibiotic treated mice would develop some increased airway inflammation when they were exposed to house dust mite extract. And they surprisingly found that it did not happen in the pups which were basically not treated with the antibiotic. And further, they found that the airway inflammation in the antibiotic treated animals was characterized by increased recruitment of eosinophils and dendritic cells in the lungs, along with increased production of airway mucus and also increased production of type 2 cytokines by the lung draining lymph node cells when they checked it ex vivo. So this was very interesting, and they tried to further, you know, uh, analyze, make more observation, and they observed that at a steady state, which they call that after 40 days of co-housing, the animals were in a steady state, that they had all the microbiota which was restored. The antibiotic treated animals showed no changes in the number of composition of immune cells as well as gut permeability. That means they were almost looking normal. But however, when they cultured the lung structural cells ex vivo along with the dust mite extract or the bacterial lipopolysaccharide, and you know that lipopolysaccharide is a pathogen associated molecular pattern of some bacteria, and it basically goes and binds to some toll like receptor, specifically the TLR4, and activates. So it induces in these animals when they cultured these structural cells and they added house dust mite extract or LPS, it will induce higher levels of various chemokines. And you know, chemokines are the ones which attract the immune cells. And it happened specifically in the antibiotic, treated mice, but not seen in the cells from non-treated mice. So that was very interesting. And in order to understand the molecular mechanisms further, they went on to conduct a single cell RNA sequencing. And you know that this is a very advanced technology where you can actually check the expression of various genes and see what is the difference between the treated animals vis-a-vis untreated animals. So they took the structural lung structural cells from both different groups of mice, and they conducted the single cell RNA sequencing uh, at the steady state, after 40 days of this, and they found that the most affected were the alveolar cells as well as the epithelial cell populations in these antibiotic treated mice. Then they did the advanced analysis of the RNA sequencing data, and they found that there were many differentially expressed genes, and among them, uh, uh, the type 2 alveolar cells, many were involved in the mitochondrial function, cellular stress, and superoxide production. And they observed that in adulthood at a steady state, the epithelial cells of antibiotic treated mice showed increased mitochondrial dysfunction, but apparently no differences in the viability, mitochondrial mass, or membrane potential, or even the superoxide production in a steady state. However, when these cells were stimulated with house dust mite, uh, and they found that it caused an increased mitochondrial mass along with membrane potential, and also increased superoxide production. And this they hypothesized that it could build up over time and cause the mitochondrial dysfunction. And further to explore the mechanisms that underlie the mitochondrial dysfunction, they used untargeted liquid chromatography mass spectrometry, the LCMS, to find out the metabolites in serum and the bronchoalveolar lavage of these mice. And they found that in serum, immediately after antibiotic treatment, there was a significant reduction in the oxygen radical scavengers. You know, oxygen radical scavengers are antioxidant mechanisms, and these are certain chemicals which our body produces, and it absorbs the oxygen radicals so that it doesn't cause any harm to the body, because all free radicals production including the oxygen-free radicals, they are basically causing a lot of ill effects because they cause the cell death and many other functions. And during any inflammation or stress, there is a lot of production of these kind of free radicals, and the oxidant radical scavengers, they basically act as antioxidants, and they absorb those. And They found that in the antibiotic treated animals, although the number of cells and everything was restored, but yet the the there was a observation that there was a significant reduction in the oxygen radical scavengers, which with most prominent one was the isopropanol. And they also found that even the precursors of isopropanol were also reduced in these antibiotic treated animals. Now they have made a observation that there are certain mitochondrial dysfunction, there is excessive production of oxygen free radicals, there is reduced oxidant radical scavengers. So in order to validate if the absence of this metabolite, which is the oxygen radical scavenger, specifically the isopropanol, could be responsible for the increased airway inflammation. So they set up the experiment, they stimulated the lung structural cells once again with the house dust mite, but now they added the isopropanol in one set of the tubes, and they found that in the tubes where there was added isopropanol, it inhibited the production of chemokine, specifically the CCL20, which is responsible for immune cell infiltration in the lungs. And it also increased the production of a cytokine IL-10, which is immunosuppressive. So isopropanol basically tried to restore the functioning, because that indicated that it was reduced, and now if you add it, it will restore the function. So they also use the to confirm their this particular finding, they also used a human airway cell line, and again they uh, you know stimulated with the house dust mite extract, and then they added the isopropanol, and they found that the addition of isopropanol would limit the production of superoxides. So very good. And finally, to prove their all these findings, they conducted an in vivo experiment in the young mice, and showed that when the young mice were given isopropanol during the antibiotic treatment, it reduced the superoxide production in the lung, and also restored the expression of several differentially expressed genes, which were earlier they found in the RNA sequencing. And these genes were specifically those which will regulate the mitochondrial function. And this indicates that the the supplementation of isopropanol during early life treatment with antibiotics effectively protected the mice uh, from airway inflammation, which was induced after 40 days of co-housing, and it reduced the immune cell lung infiltrates, and also the levels of cytokines and chemokines. So guys, what is the carry home message? The carry home message is that use of antibiotics in the early life in children causes development of allergies and asthma later in their life. That is the one message. And these authors have shown that this happens mainly because of the depletion of normal microflora in the gut, which leads to the depletion of certain specific metabolites, which include the oxygen radical scavengers, and one of them is the isopropanol. And then this study further elaborates the crucial gut lung connection. Gut lung connection is basically the connection between the microflora of these two major organs in the immune health, that it has a major connection with that. And the authors finally suggest that a possible adjunct therapy uh, when the antibiotics has to be used in the early life in children, so that the adjunct therapy will protect them for future allergies and asthma. So I'll close down here, and I'll let you guys go with the suggestion that you should go and read the papers and the literature I have cited, and keep listening to the podcast, and send me suggestions once in a while, in the comment box, so that I can bring out more interesting case, studies, and discuss with you guys. Till then, thank you.